Myself, Madhusudhan, working as senior research fellow from India and from Bangalore University. As we all know, there are many plant diversity hotspots around the world. In India, Western Ghats and Himalayan region together is one amongst the important biodiversity hotspots. Western Ghats in India is a very beautiful forest range where it has dense evergreen forests to the grasslands and deciduous forests. In these forests, Calamus plays an important role as it makes a biological defense to protect the other plants and animals which are very sensitive in the forest. However, in recent past, with the constant increase in the population, these defenses have been breached and plants have been exploited for economic values, and this has resulted in many plants being pushed to endangered list. In Western Ghats, there are roughly around 45 species of Calamus, and more than 10 species are considered as endangered, and one species, that is Calamus roxburghi, is feared to be extinct. With these reasons, the work to conserve these genera was selected, and few important species were selected for the conservation. Now, I'll be speaking of one such plant, that is Calamus huegelianus. It is a very beautiful plant, slender climber. It belongs to family Ericaceae, and it is considered as a non-timber forest produce. Calamus huegelianus is a high climbing, as I told, it's a climber, clustering canes. It forms a cluster with a diameter, the stem diameter from up to 3 centimeters to 5 centimeters along the sheath, and without the sheath it can be maximum of 2.5 centimeters. It is economically important taxa, as I mentioned, endemic to Western Ghats. This genus Calamus distributed in India, as I told, in Western Ghats, Eastern Himalayan region, Andaman Nicobar Islands, and few other forests in India. This is a spiny climber. Plant is characterized by beautiful feather-like leaves. The plant needs absolute shade in its early development, and as it grows, it reaches to the sky, competing for the sun. The stem is the main part economically, and it's commonly called as a cane or a rattan. It's commonly used in making of many household articles like chairs, swings, and many other baskets, many things. For many families which are living around, surrounding the forest, these products, other than the timber, these plants are the livelihood for them, and thousands of families have been depending on these plants. And because of various reasons, like deforestation, urbanization, unscientific collection of these plants have depleted the number of plants, and there is a need for conservation. So the Calamus, why it should be conserved or mass propagated? There is an immense imbalance between demand and supply. As we know, like in many furniture industries, many furnitures which are made by nice thick colorful timber, this gives a very potent competition where it has a very good property of taking up the polish, durability, elasticity, and many other characters are attributed for this plant. Indiscriminate extraction of plant, rattans. That is because of the growing demand for these plants. People are extracting these plants from wild indiscriminately, and these plants are monoecious, that is male and female plants separate, and these plants will take at least 8 years for producing flowers. So people who are living surrounding the forest just extract whenever they need the plant, and like this, they are like if a male plants are cut down, there is a lack of pollination and the productivity is harmed. And percent seed viability and germination is very low in many of the species. Seeds do not germinate if stored for a long time because the hydration in the seeds is very sensitive and the seeds are very sensitive for drying. Vegetative propagation for mass multiplication is limited as the suckers or the other vegetative multiple parts are very sensitive and the survival of these suckers are very low. With this background, we had framed two experiments, that is study of seed germination first and then the plant tissue culture work. So this is a beautiful state where I come from, Karnataka, and these plants were collected in that marked region, that is Chikmagalur, Hassan and Dakshin Kannada districts. Mainly the fruits and the shoot tips, seedlings were collected. First, I'll be speaking about the seed germination part. The fruits were soaked in water for 24 hours and rubbed against each other to remove all the outer sarcoptesta and the outer layer. Sorry. The seeds were thoroughly washed with soap, P20, in running water for 30 minutes to enhance the adhesion capacity of the seeds, and then surface sterilized by bevestin, a fungicide, for 30 minutes. The seeds were then washed in tap water to remove all the harmful fungicide. And pre-treatments were given. That is, one is the seeds were treated with GA3 at 123 mg per liter concentration. Acid treatment, the seeds were treated with HCl at 1, 5 and 10% acid concentration. Boiling water, like the seeds were placed in a bag and kept in boiling water for different time durations, and untreated seeds were considered as control. 4 experiments were selected for the seed germination. That is, one is in vitro germination studies, that is the tissue culture method. The second, paper bridge method, desiccator method, germination on different substrate. That is the raw data what I collected from the forest people were like they were using cow dung rock, fresh cow dung for germinating these plants and different other materials. So I tried out many other substrates to see the effect of the substrate on the germination. These were the 5 species which I had collected. The first one being Calamus prasinus, Thwatesi, huegelianus, Nagabetoi and Vataila. This is like a paper bridge method where blotting paper was taken and made a cup shaped and like a bridge which is connected to the sterile water, and seeds in each tube only one seed was placed, and seeds had been germinated. And this is the seeds when germinated in different substrates. And observations were made for mean germination time, seedling vigor, and germination percentage. So the average of the seed germination were calculated for seed germination percentage. For mean germination, mgt equals sigma nd by n. This was the formula which was followed, and seedling vigor, again the formula which is mentioned was followed. This is the table where the cow dung and cow dung mixture, as it was mentioned by the forest people, showed the maximum percentage of germination, that is 43.3 plants per 100 seeds. And other substrates were being soil alone, sand, like compost alone, compost mixture. Mixture here means 1:1 ratio with compost, soil and sand, and all these were sterilized. fresh cow dung and the mixture, sheep dung, cow dung, ash, and perlite and vermiculite were also used. So here we can see the cow dung and cow dung mixture had shown maximum number of seed germination results. Though the best germination percentage were seen in vitro techniques, that is the tissue culture techniques, the seeds, as the seeds were collected from the forest floor, I actually think that they had lot of fungal spores inside that, as the sarcoptesta is very sweet, and the contamination was very in such a way that it was not able to control, because any more treating with the acids or anything was lethal for the embryo. Desiccator method and paper ridge method both showed results with the highest vigor, but again they were like in this particular species the fungal intrusion was high. As a whole, sheep dung manure and cow dung manure were the best substrates for growing these plants, which had the maximum germination percentage, vigor and the vigor and the mean germination time, that is the time of germination. These plants have taken 110 days an average for germination. With this background, that is, as I had tried these experiments in 5 species of Calamus, and 3 species showed less percentage of germination and high time duration for germination. One among those was this Calamus huegelianus, Nagapetai, and Prasinus. The other 2 species, that is the Calamus Vataila and Twetesi, had shown 90% of seed germination, and all the parameters was very good. So we neglected those 2 plants for tissue culture work, and we had selected these 3, and I will be talking about only huegelianus here. As I told, the material methods remains the same. The plants were collected from the forest, and leaf explants, root tip explants, and shoot tip explants were used for tissue culture, in where only shoot tip cultures were shown response, and other 2 were didn't show any response for this experiment. They were thoroughly washed as the customary tissue culture techniques were followed. Surface sterilizing and media preparation was done, and 2 media were selected, that is MS media and L2 media. Both mediums were supplemented with different hormones or the plant growth regulators at in different concentrations, singly or in combinations. Sucrose was 3% and agar 8.8%. Again, the cultures were incubated in a culture room. Both indirect and direct organogenesis was obtained depending on the type of concentrations of the hormones supplemented to the media. L2 media supplemented with 2,4-D, 2,4-D and IAA along with BAP were proved to be the best combination of hormones, where direct and indirect regeneration were shown with 0.9 standard error. These were the results. This is direct regeneration of shoots. The results are there in the media where IAA, NAA and BAP were shown, where the hormone is treated with as single. These were again with treatment of BAP, NAA, BAP 2,4-D and IAA to BAP. And this is at 0.5 auxin plus 2 mg per liter cytokinin. Rooting acclimatization, which is a very important step in tissue culture, was carried out. The multiple shoots that were obtained through in vitro cultures were transferred to rooting mediums with NAA and IAA. After 45 to 50 days, the rooted plants were transferred to plastic bags containing vermiculite and kept in polyhouse condition. Thus, hardened plants were transferred to pots containing soil, sand, and manure. 3 months old seedlings were transferred to field, and there were 40% of survival rate. This is rooting of these plants in vitro. So, conclusion. Today, the conservation of the phytodiversity in the order of the day. As the ever increasing pressure on forests because of urbanization and encroachment of forests for farming and urbanization is on a high. The above mentioned protocol helps to generate large number of saplings in short time and also reduces the pressure on wild stock. And this the protocol can be very efficient in increasing the production of cane in meeting the demand, which will mainly help in upgrading the rural economy.